So for this video, I'm going to talk about gene regulation and how apoptosis is initiated. So I'll start talking about 3 different proteins, and then from there I'll add in a couple others that kind of regulate the 3. So the first 2 we've heard of before, that's P53. And so P53, it has 2 functions. It can halt cell division, and it can activate apoptosis, but it doesn't necessarily have to activate apoptosis, only at high concentrations, so when it's really high. At lower concentrations, it can just halt cell division until there's a better time when the cell can divide. Then the other 2 proteins are retroblastoma protein, RB, and also EF2. And so basically, retinoblastoma protein is usually phosphorylated. Okay, and both of these initiate cell division. Okay, and so what usually happens is this is phosphorylated, freeing up EF2. So EF2 is a transcription factor for cell division. Okay, so P53 is usually expressed in low levels, but it's regulated by a protein MDM2, which has 2 functions. It binds to P53, which prevents it from actually initiating transcription of apoptosis, like the genes for apoptosis, and it also transports it out of the nucleus, where it then actually helps it get degraded. Okay, but in cases of DNA damage, P53 not only becomes upregulated, so it'll be transcribed more, it also becomes phosphorylated, and when it phosphorylates, it can't be bound by MDM2. Now that it's activated, it can bind to and transcribe apoptotic genes, and it also, through a step where it activates P21, which then inhibits cyclin-dependent kinases. And when these cyclin-dependent kinases are inhibited, they don't phosphorylate RB. So now that RB is dephosphorylated, it actually sequesters EF2. And so now EF2 can't work as a transcription factor to initiate cell division. And so basically, cell division is halted.